Okay, so welcome back to this next video in which we are discussing calcium signaling. Okay, right, so far we've looked at the mechanisms used to maintain a low calcium concentration in the cytoplasm of the cell and a high calcium concentration in the endoplasmic reticulum of the cell, or sarcoplasmic reticulum of the cell if we're talking about a muscle cell. Okay, what we now want to start looking at is calcium signaling. Okay, so we want to look at how we can generate calcium signals within the cytoplasm. And one of the key ways that we can generate calcium signals in the cytoplasm is by releasing calcium from the endoplasmic reticulum. And one of the ways that we can release calcium from the endoplasmic reticulum is through calcium channels that are called IP3 receptors. Okay, and it's signaling with the IP3 receptors that I want to turn my attention to now. Okay, right. So the first thing that we need to discuss if we are going to discuss IP3 receptors is the generation of IP3. Okay, so IP3 is crucial in the opening of IP3 receptors. It's not a direct mechanism. IP3 doesn't directly open the IP3 receptors. It's more subtle than that. Okay, and we'll come to that later on. But for now, what we want to talk about is how do you actually create this molecule called IP3, which is responsible for the opening of the IP3 receptors. Okay, so basically, the way IP3 is produced is by the breakdown of a phospholipid that is present within the cell membrane. Okay, and this breakdown is going to be catalyzed by a bunch of enzymes that are collectively called phospholipase C enzymes. So PLC here stands for phospholipase C enzymes. Okay, and there are a whole bunch of phospholipase C enzymes which are all regulated in different ways, and we'll talk later on about the regulation of the phospholipase C beta and gamma families. Okay, but for now we want to look at the thing which unifies all phospholipase C's together, which is what do they actually do. Okay, and what they do is they break down a membrane phospholipid called PI45P2, or sometimes just called PIP2. Okay, strictly speaking, it should be abbreviated to PI45P2, but people often just abbreviate this down to PIP2, and you can also hear it referred to as just PIP2. Okay, they break down this membrane phospholipid to produce IP3 and diacylglycerol. So the first thing I want to discuss is what is this membrane phospholipid? Okay, so let's see what the full name of this molecule is then. Okay, so what does PI45P2 stand for? Well, the first P stands for phosphatidyl. Okay, so that's the first P here. Then the I is for inositol. Okay, the 4,5 is for 4,5. Okay, and then the P2 on the end here means bisphosphate. Okay, so the P is for phosphate, and then you've got two phosphate groups, so it's bisphosphate, or P2. Okay, so that is the full name for for PIP2 or PI45P2, whichever abbreviation you prefer to use. If you just abbreviate it down to PIP2, people will know what you mean, okay? Because if you don't put numbers in the middle to say which carbons of the inositol ring the phosphate groups come off, then people will assume that they're coming off the 4th and the 5th, okay? But the more correct abbreviation is PI45P2, like so. Okay, right, so let me discuss what the structure of this molecule actually is. So I've told you that it's a membrane phospholipid. It's a constituent of the phospholipid bilayer. It's one of the things out of which the phospholipid bilayer of the cell membrane is made. However, it's not a particularly common phospholipid. Okay, it's quite a rare one, but it is normally present in the plasma membrane. Okay, so let me show you a cartoon then for the structure of this molecule. Okay, right, so if we're going to discuss the structure of this molecule then, we firstly need to discuss this prefix, phosphatidyl. Okay, so we're going to firstly take on this portion of the name, the phosphatidyl prefix here. What does that actually mean? Okay, well that means that the entire molecule is going to be based on another molecule called phosphatidic acid. So before we can discuss phosphatidic 4,5-bisphosphate, we firstly need to discuss what is phosphatidic acid. Okay, so phosphatidic acid is not just one molecule, okay, it's a whole bunch of molecules, as you'll see in a moment, okay, and all of these molecules are, as their foundational structure, they are based on glycerol. Glycerol is the foundations for a phosphatidic acid molecule. So let me draw a phosphatidic acid molecule here, okay, so this is what's going to represent the glycerol, and I'll just draw it out and then I'll explain the different portions to you. Okay, so this is my cartoon for a phosphatidic acid molecule. So let me now highlight up the different portions and explain what they're representing. So firstly, we'll start off with this vertical line here that I've now coloured in green. Okay, so this is representing glycerol. Okay, now the proper name for glycerol, the chemist's name for glycerol rather than the biochemist's name, is propane 1,2,3-triol. Okay, and although propane 1,2,3-triol is more of a mouthful than glycerol, it's a more useful name because it tells us exactly what we're dealing with here. It tells us that we're dealing with a 3-carbon molecule, that's the propane, where you have alcohol groups coming off the first, the second, and the third carbons. Okay, so if you like, this portion here is representing carbon number 1 of the glycerol molecule, this portion here carbon number 2, and this portion here carbon number 3. Okay, and every single one of those carbons has a single alcohol group coming off it. Okay, now these two horizontal lines here, which I will colour in orange, okay, these are representing fatty acids. Okay, now fatty acids are also called long-chain carboxylic acids. Okay, and again, that name gives us more insight into what the chemical structure of these things actually is. Okay, it tells us that these are carboxylic acid molecules which have really long tails, basically. Okay, and in fact, their really long tails are going to be really hydrophobic, so they're not going to interact well with water at all. Okay, so this is the lipid part of this structure. Okay, so off the glycerol molecule, we have attached onto the alcohol groups that come off the first and the second carbons of the glycerol molecule these long-chain carboxylic acids by ester links, basically. Okay, so that's the portion that we've discussed so far. Okay, now phosphatidic acid is an example of a phospholipid, okay, and indeed phosphatidic 4,5-bisphosphate, which is derived from phosphatidic acid, is also going to be a phospholipid because it contains a phosphatidic acid molecule. Okay, now what is the definition of a phospholipid? What do you have to actually meet to be considered a phospholipid? What are the criteria here? Okay, well the criteria are two. You have to have a lipid component within your structure, okay, and you also have to have a phosphate group within your structure. Okay, so this phosphatidic acid molecule certainly meets both of those criteria. Okay, we are now looking at the lipid structure here, okay, and we're about to see the phosphate group. Okay, so if this is going to be a phospholipid, it has to have a phosphate group somewhere, and this is the phosphate group here, attached to the alcohol group that comes off the third carbon of the glycerol molecule. Okay, so this is a phosphate group. Okay, and that structure that we've now been through, that is a phosphatidic acid molecule. Okay, glycerol with two long-chain carboxylic acids linked here and here. Okay, and then off the alcohol group of the third carbon of the glycerol molecule, you have this phosphate group here. So phosphatidic acid molecules are phospholipids, okay, and all the things that are derived from phosphatidic acid, including this molecule that we are hunting for, which is phosphatidic 4,5-bisphosphate, they are going to be phospholipids because they contain phosphatidic acid. Okay, so I said that there wasn't just one phosphatidic acid molecule, and there isn't. And the reason that there is not just one phosphatidic acid molecule is because I haven't specified what these long-chain carboxylic acids should be here. Okay, you can put many different long-chain carboxylic acids in these two slots, and they don't need to be the same long-chain carboxylic acid. So that means that there is a huge number of different molecules that are all considered phosphatidic acid, basically. Okay, right. So how then now do we go on to build phosphatidylinositol 4,5-bisphosphate? Well, this phosphatidyl prefix in this name means that the molecule is going to contain phosphatidic acid. Okay, so how do we turn a phosphatidic acid molecule into a phosphatidylinositol molecule? Well, basically what we now have to add on to this is an inositol ring. So let me just discuss what inositol is then. Okay, so let me give you the correct, or the more official now, name for inositol. So inositol, like glycerol, like fatty acids, they're the old names for these. Okay, they now have more correct names, okay, and more useful names. So inositol's more useful name is cyclohexane 1,2,3,4,5,6 hexol. Okay, so cyclohexane 1,2,3,4,5,6, and then hexol. Okay, and again, this might be a nightmare of a name, but it's a very useful name. It tells us exactly now what inositol actually is. Okay, so let me talk you through this name then. So cyclohexane. Okay, cyclohexane just means a 6-carbon molecule. That's what hexane is. Okay, where it's now going in a cycle. So let me draw this with skeletal formula down here. Okay, so remember, in skeletal formulae, you don't show carbon atoms. They're implicitly shown by corners, and you don't show hydrogen atoms coming off carbon atoms. Where you have missing bonds coming off carbon atoms, those are assumed to be hydrogen atoms. Okay, so that means cyclohexane can be drawn incredibly simply, just as a hexagon like so. Okay, because we have 6 carbons, 1, 2, 3, 4, 5, 6. Okay, and now all of the bonds between the carbons in cyclohexane are going to be single bonds, so all of the other atoms coming off those carbons, okay, and each carbon needs 2 atoms coming off it, are going to be hydrogen atoms, basically. So that's pure cyclohexane. Of course, we don't want pure cyclohexane. We want cyclohexane 1, 2, 3, 4, 5, 6 hexol. Okay, so we want to put alcohol groups of every single one of those carbons. So at present in cyclohexane, every single one of these carbons has 2 hydrogens coming off it. We want to take off one of those hydrogens and replace it with an alcohol group for every single one of those 6 carbons. Okay, right. Now, this may seem incredibly simple, however there is a subtlety here that we need to dwell on, okay, because if you want to understand why PI45P2 is called PI45P2, then we need to understand something about inositol. Okay, if we specifically want to understand the 4,5 bit, we need to understand something about inositol. Okay, and this subtlety here is optical isomerism. Okay, so let me explain why inositol is going to have multiple optical isomers. So if you imagine that all these 6 carbon atoms are sitting beautifully in the plane of the piece of paper, okay, then how will the 2 other bonds of each of those carbon atoms be oriented? Okay, well, if we look at this carbon here as an example, one of these bonds will be coming out of the page at that sort of angle that my pen is on at the moment. Okay, and the other bond will be going into the page, sort of like the mirror image of the way this one's coming out, basically, like in that sort of a direction there. Okay, so every carbon of the inositol ring is going to have an alcohol group coming off it and a hydrogen coming off it. which one's going to come out of the page and which one's going to go into the page? That's the question now that gives rise to the different optical isomers of inositol. Okay, so to show you some extreme examples, okay, we could have all 6 alcohol groups coming out of the page towards us, i.e. on the same side, and then all of the hydrogen atoms going into the page away from us, i.e. on the same side. So that's one optical isomer of inositol, but of course there are other optical isomers which wouldn't have all 6 alcohol groups on the same side. Okay, and it turns out that if you go through all the plausible optical isomers of inositol, it turns out that there are 9 in total, so 9 optical isomers of inositol. Okay, now, luckily for us, in biology, there is only one that is actually used. Okay, so in biological systems, in cells, they like one form of inositol, and that's the only one that they use. Okay, and this specific optical isomer of inositol that's used within cells is called myo-inositol. Okay, and I'm now going to show you the structure of myo-inositol, and I promise you there will be some relevance to knowing this in the end. Okay, right, so let me show you how myo-inositol's alcohol and hydrogens are oriented coming off the carbons. Okay, so we'll start with this carbon here. So this carbon's alcohol group is going to be coming out of the page towards us rather. Okay, so the way I show bonds coming out of the page towards us is in this way, okay, the bond getting thicker towards the end. Okay, so the alcohol group will be coming out of the page towards us, and that means that the hydrogen coming off that carbon will be going into the page away from us. Now, of course, I don't need to show that because we're drawing a skeletal formula. Okay, right, but it is there, be aware of that. Okay, so, next carbon, second carbon, it's going to be exactly the same case. The alcohol group of this carbon will be coming out of the page towards us, and the hydrogen will then be going into the page away from us. Third carbon, it's the same again, okay? The alcohol group will be coming out of the page towards us, and the hydrogen will be going into the page away from us. Now it's suddenly going to change. The alcohol group coming off this fourth carbon is going to go into the page away from us, and the way I show bonds going into the page away from us is with this dashed line like so. And that means now that the hydrogen coming off this carbon will be coming out of the page towards us, but again, of course, we won't show that. Okay, then off the fifth carbon it goes back to being the case that all the alcohol group is coming out of the page towards us, and the hydrogen is going into the page away from us. Okay, and then finally on the sixth carbon over here, the alcohol group goes into the page away from us. Okay, so that is the specific orientation of the alcohol groups coming off the carbons of the myo-inositol ring, basically. Okay, right. Now the carbons of the myo-inositol ring are set in stone as far as their numbering is concerned. Okay, so chemists have named the carbons of a myo-inositol ring as follows. Okay, so this carbon here is carbon number 1. This carbon here is carbon number 2. This carbon here is carbon number 3. This one is carbon number 4. This one is carbon number 5. And this one is carbon number 6. And we will be using this in just a moment. Okay, right, so that is the structure of myo-inositol, the optical isomer of inositol that is found within cells, basically. Okay, so that is myo-inositol here. Right, okay, so now let's build phosphatidic we will be using myo-inositol, but I will be dropping the myo from now on. Okay, whenever I refer to inositol from now on, I mean myo-inositol. I don't mean any other form of inositol. Okay, I mean the biologically found inositol. Okay, so we're now going to attach on then an inositol ring onto my phosphatidic acid molecule. Okay, so how are we going to do this? Well, let me firstly just redraw out my phosphatidic acid molecule here. Okay, so I'll just colour in the different portions here. So here is the glycerol in green there. Here are the 2 long chain carboxylic acids attached to the first and second carbon's alcohol groups. Okay, and here then is the phosphate group coloured in purple coming off the alcohol group at the third carbon of the glycerol ring. Okay, now what we're going to do is we're going to attach the inositol ring here onto the phosphate group of a phosphatidic acid molecule. Specifically, we're going to attach the alcohol group coming off the first carbon of this inositol ring onto this phosphate group. Now I'm going to show this simply like so. Okay, I'm going to represent the inositol ring now just as a blue hexagon. Okay, so here now is my inositol ring. Okay, but remember it's the first carbon that is being attached to the phosphate group there via its alcohol group. Okay, right, so that molecule that we've now created is phosphatidyl inositol, and it's still a phospholipid because it contains the phosphatidic acid molecule, which had the two features that are needed to satisfy the definition of a phospholipid. Okay, right, now we don't want phosphatidyl inositol, we want phosphatidyl inositol 4,5-bisphosphate, so now we need to add phosphate groups coming off the fourth carbon of the inositol ring's alcohol group and the fifth carbon of the inositol ring's alcohol group. Okay, and we will be now just adding them on to the fourth and the fifth carbon, which is going to be this carbon here and this carbon here. Okay, like so. So the phosphate groups will be attached onto those alcohol groups via phosphoester links. Okay, right. Now, this is why I wanted to go into the detail of what myo-inositol actually is, because if I had just drawn this cartoon, you would have said, well, why is that not called phosphatidyl inositol 3,4-bisphosphate? Why do we not call that one the third carbon? Okay, if this is the first one, why don't we count around this way? 2, 3, 4. Okay, so it should be phosphatidyl inositol 3,4-bisphosphate. But I hope you now understand why that truly is phosphatidyl inositol 4,5-bisphosphate. It's because the third and the fifth carbons of myo-inositol are not equivalent, even though in my simple cartoon here, this carbon is equivalent to this one. Okay, in reality, they're not equivalent. The third and the fifth carbon are different. Okay, they're not symmetrical, because look, this molecule is not symmetrical. If I take that as the line of symmetry, it's not symmetrical. This one is differently oriented to this one, which means that the fifth carbon is different from the third carbon. You can tell that this is a different carbon to this one, basically. There isn't that symmetry, okay? So the third and the fifth carbons of the myo-inositol ring are different. Okay, to really labour this point, the fifth carbon here is between two carbons which have their alcohol groups oriented in the opposite direction to the way that the fifth carbon's alcohol group is oriented. Whereas the third carbon is between two carbons, one of which has its alcohol group oriented in the opposite direction to the way it has its oriented, but then the other has its alcohol group oriented in the same direction as it has its alcohol group oriented in. Okay, so the fifth and the third carbon are distinguishable, and it is the fifth carbon's alcohol group that we have added a phosphate group onto in phosphatidyl inositol 4,5-bisphosphate. So that is why this molecule is PI45P2. Okay, right. So that's an introduction then to PI45P2. What we now want to see then is what phospholipase C enzymes are actually going to do to PI45P2. Okay, what are they going to break it down into? Okay, so let me draw the cell membrane here. Okay, now put a PI45P2 molecule into the cell membrane here. Okay, so I'm just copying this molecule out into a phospholipid bilayer like so. So here is the phosphatidic acid molecule, here is the inositol ring here, okay, and then we've got a phosphate group coming off the fourth and the fifth carbon of the inositol ring's alcohol groups. Okay, so let's just colour it in once again. So here is the glycerol in green there. Okay, here are the long chain carboxylic acids in orange. Okay, here is the myo-inositol ring in blue. Okay, and here is the free phosphate groups in purple. Okay, right, so So that is our PI45P2 molecule sitting in the phospholipid bilayer, basically, with its head here, which is the polar portion, dangling into the cytoplasm. So here is PI45P2 sitting in the cell membrane. Okay, so as I said at the start, it is a normal component of the cell membrane. You do have PI45P2 in your cell membranes. Okay, however, it's a rare component. It's not a common component. Okay, it's difficult to come by. Okay, now, the characteristic feature that links all of these phospholipase C enzymes, okay, is that they can all break down PI45P2, and the way that they break down PI45P2 is by cutting this bond here between the phosphate group here and the alcohol group at the third carbon of the glycerol molecule, like so. Okay, now what two products will you then produce from this? Well, you'll produce one product that's going to remain in the phospholipid bilayer because it's anchored there by its lipid groups. Okay, and that is a glycerol molecule with two long-chain carboxylic acids attached. Let me just show this here. There is the glycerol molecule in green, and here are the two long-chain carboxylic acids attached. Now that molecule is called diacylglycerol. Okay, or it's rather an example of a diacylglycerol. After all, we don't know what the two long-chain carboxylic acids are, so it's a massive, great class of molecules rather than a specific molecule. Okay, so this is a diacylglycerol, also called a diglyceride. Okay, right. So here is our diacylglycerol, one of our products of this breakdown by phospholipase C. Okay, and that will have important signaling properties later. We're not going to study the signaling properties of diacylglycerol at the moment, but we will come back onto it when we want to discuss protein kinase C activation. Okay, the other product of this cleavage by phospholipase C enzymes is this portion here, the myo-inositol ring, like so, with three phosphate groups attached to it. A phosphate group coming off the alcohol group of carbon number 1, a phosphate group coming off the alcohol group of carbon number 4, and a phosphate group coming off the alcohol group of carbon number 5. Okay, right, so let's colour this in. Here is the inositol ring in blue here. Okay, and here are the phosphate groups coming off carbon number 1, 4, and 5 here. Okay, so this molecule is called inositol 1,4,5-trisphosphate. Okay, so inositol, that's the ring here, and in fact some people will even call this myo-inositol 1,4,5-trisphosphate. It's very rare to see someone actually call it myo-inositol 1,4,5-trisphosphate, but if you see it, know what that means, don't be scared by it, and know that it's just someone being extra cautious. Okay, but generally people don't bother now specifically specifying the fact that this is myo-inositol. Okay, it's taken as understood that it is myo-inositol. So inositol 1,4,5, because those are the carbons off which you have the phosphate groups, and then tris for free phosphate. Okay, so inositol 1,4,5-trisphosphate, and for short, inositol 1,4,5-trisphosphate is abbreviated down to I for inositol, P for phosphate, and then we've got three phosphate groups coming off it, so it's IP3. Okay, right, so this is the IP3 molecule here. Okay, so basically, phospholipase C enzymes then, they break down PI45P2 molecules, this normal component of the phospholipid bilayer, into these two molecules, diacylglycerol and IP3. Okay, we will come back to diacylglycerol. For now, IP3 is the one that we're going to be incredibly interested in, because this is the one that's going to go off and act on the IP3 receptors. Okay, right, so we'll call it there for this video. In the next video, what we will talk about is the different types of phospholipase C enzymes, because we've talked now about what phospholipase C enzymes do. We've talked about how they break down PI45P2 into diacylglycerol and IP3, IP3. Okay, what we now want to see is how do you activate phospholipase C enzymes, because they're not normally active. Okay, you have to activate them in some way, and this is where different families of phospholipase C enzymes have very different means of regulation. Now, we're not going to go through all the different families of phospholipase C enzymes. We're going to go through the two most important, okay, which are the phospholipase C beta family, which are the ones which can be activated by the famous G protein cascade, okay, and the phospholipase C gamma enzymes, which can be activated by receptor tyrosine kinases. Okay, so in the next video we'll look at the activation of phospholipase C enzymes of different families by those two very different receptor types, okay, and then we'll go on to how the IP3 is then going to act on the IP3 receptors.